Greetings everyone. In this video we are going to discuss about our new topic, which is the biochemistry. Previously we are done with all of our organic chemistry topics, and in this video we are going to have a detailed understanding of how chemical reactions occur in living systems. So that's going to be again very exciting. And before discussing that, we are importantly going to discuss something that's going to help us in the industrial chemistry chapter. The thing which we are going to discuss over here is this: polymers. We need to first gain a basic understanding of what polymers are, and then we are going to get into the macromolecules, which are carbohydrates, proteins, lipids. So first, when it comes to macromolecules, they are broadly classified into two types, which is organic and inorganic. Organic ones are giant structures like diamond, graphite, and sand, and organic ones involve two further types, which includes biopolymers, which are found in living systems, lipid, protein, carbohydrates, and nucleic acids, and those which are made synthetically, known as synthetic polymers, such as plastics, synthetic fibers, and rubber. Now let's just move on to a basic understanding of what a polymer is. Whenever we are going to have a very small, a very small repetitive a unit which is repeating continuously, chemical unit, known as monomer, to form a long chain to form a long chain called polymers. It's that when we are having a very very small repetitive chemical units called monomers, which form a long chain called a polymer. Now when we talk about polymers, they are of three major structures. Number one structure of polymer is linear polymer. That's when the chain is made in a single direction. When the chain is made straight without any branches, it is a linear polymer. And when the chain is made with branches, such as it is having branching over here, so it's going to be a branched linear polymer. When there are multiple linear chains which are connected with each other through some cross bridges, then it is known as cross bridge linear polymer or interconnected polymer. So these are major structures. Now let's just understand a few types of polymers as well. So broadly we can classify polymers into three types. Number one type which you're going to understand is homopolymer. Homopolymer is one in which the monomer which we are going to use to make the polymer is of the same type of monomer, right? Same type of monomer is used. For example, if I take acetic acid, I take acetate, a vinyl acetate actually. Let's say I take vinyl acetate, right? And I what I'm going to do is I'm going to polymerize vinyl acetate, which will form polyvinyl acetate. Polyvinyl acetate. So in this I'm going to use only one monomer, which is vinyl acetate, and then there's an important thing that I'd like to share about, which is heteropolymer. Heteropolymer, which is basically further divided into two types, which is copolymer and terpolymer. Let's just understand what a copolymer is. Copolymer is the one in which we are having two different types of monomers, which is joined together. For example, when we are going to have dicarboxylic acids, we're reacting with alcohols, then we are going to produce a monomer, which is a copolymer. Another example is, let's say if I take multiple carboxylic acids with multiple alcohols, then it's going to produce a copolymer. And then we have terpolymers. Polyesters are examples of copolymers. Polyesters. And on the other hand side, we have terpolymers. Terpolymers are having three monomers in them. A very famous example is the acrylic resin. Acrylic resin basically is the paint which we use for car industries, which we use in car industries. The paint of car which is used in car industries is very weather resistant, and that is because of the strong abrasive resistance and chemical resistance in it. So one thing which is very important is its components. Its components are acrylic acid, meth acrylate, methyl meth acrylate, and then we have butyl acrylate. These are some important components. And there's one more thing, that it is closely associated with acrylonitrile, and that basically forms acrylic resin. Now, if you just see this is a tertiary polymer because it includes three different monomers. Now, it is further divided into two types, which is number one, thermosetting polymer, and second type, which is thermoplastic polymer. What is a thermoplastic polymer? Whenever we are going to have a tertiary polymer which can be softened upon heating and which can be hardened upon cooling, then that is known as thermoplastic polymer. Example of this can be plastics, toys, and pipes. All of these are thermoplastic polymers. When it comes to thermosetting polymers, these are those polymers which cannot be softened upon heating. Rather, when we are going to heat them at a very high temperature, they will decompose rather than melting, and they are not going to harden on cooling as well. Example of this is synthetic varnishes, and epoxy resins. This is important thing. Now let's just move on to another important part, which is about the polymerization process. How does the polymerization happens? So there are two major ways in which the polymerization happens. Number one way is through free radical substitution. When we are going to perform polymerization through a free radical reaction, through free radical addition reaction, then at that point this is known as a free radical addition polymerization in simple words. So what actually we do is we provide heat or photochemical radiations to a peroxy acid, due to which it generates free radicals, and those free radicals then again go through the process of propagation. They are again the three steps: initiation, propagation, and termination, through which the entire procedure happens. This is the free radical mechanism, which is for this is a free radical mechanism which is for vinyl benzene. On the other side, we have another mechanism which we are known as condensation polymerization. Condensation polymerization is the one in which a water molecule or methanol molecule or an ammonia molecule is released, and that basically causes to form a bond, an ionic bond. An ionic bond. Generally, the bond is ionic in nature, but there can be some exceptions as well. So that reaction is known as condensation reaction because there's a release of a condensate. There's a release of a condensate, which is H2O, or it is, let's say, ammonia, or it can also be methanol. So if you just look into this structure, we are going to, this is basically the mechanism for polyester formation. What actually I do is I take a dicarboxylic acid, right? I take a dicarboxylic acid that it is having it is basically a dicarboxylic acid which is reacting with a dihydric alcohol, due to which it forms a polyester. That is done through this mechanism. Now let's just look into some synthetic polymers, some common reactions for synthetic polymers. The first one is polyvinyl chloride. It is an addition reaction again, and that's again, as I previously mentioned, it is addition reaction and it is copolymer. Sorry, it is addition reaction and it is homopolymer. And when you are going to look into it, you'll actually find the thing that polyvinyl chloride is made in the presence of few conditions, such as 52 centigrade is required, 9 atm pressure is required, and usually we do not have a catalyst, but we can add any plasticide in order to improve its flexibility, in order to make the compound pretty flexible. And alongside that, the uses are pretty clear. We use it for making pipes, for making gramophone recorders. We use it for making um, you know for making pipes, for some and for making floors, and in that way it is pretty useful. On the other side, we have very famous compound which is known as styrene. Styrene basically is the this is the same reaction we looked into previously. This is the addition reaction. Again, it's the addition polymerization through free radical addition polymerization, and that is again a homopolymer. In this reaction, we actually get a polystyrene. Polystyrene is used for decorative purposes, for all the packaging of material, and the cosmetic bottles, the glass containers, as well as there's toys which are having polystyrene covering, the shiny covering over them. All right. Then we have another reaction. This is one which we looked in right now. This is what we call as a polyamide resin. Polyamide resin is basically the most famous type of resin which we have in society nowadays. A polyamide resin, what we actually do is we take a dicarboxylic acid with a diamine, and then we take a polyamine and we take a polycarboxylic acid, and when we are going to perform a condensation reaction, we will get a polyamide resin. That polyamide resin is known for its strength, toughness, rigidity, and elasticity, and chemical resistance. And one famous type of and this is a process through which we make nylon. Polyamide resin is also known as nylon. The most famous type of nylon is nylon 66. This is made through adipic acid. Adipic acid is have hexanoic acid, hexane dioic acid, and hexamethylene diamine, such that when they are heated up, they are going to produce nylon 66. Why do we name it nylon 66? Because both adipic acid and hexamethyldiamine are having 6 number of carbon atoms. So we name this as nylon 66, and it's having a lot of uses. It is used for making a lot of different products in our industry. On the other side, we have epoxy resins. Epoxy resins are basically formed because of the epoxy group which is present. This is basically an ether group, but we also call this as epoxy group. So, epoxy group, when particularly epichlorohydrin reacts with diphenyl all propane, it's actually going to form an epoxy resin. That epoxy resin is highly resistant to oxidization, and it is highly resistant to chemical attack, and it is very tough, very tough, and very adhesive. And it is because of these properties we use it for painting of industrial material, industrial machinery, and industrial material. Hydropower plants, water stations, dams, bridges are all painted with epoxy resins. So these are some of the important things about compounds. All right, there's one more important thing that we previously discussed. It's again the acrylic resin. Acrylic resin is into having acrylic acid, methyl acrylate, and we have butyl acrylate, acrylonitrile, which are used to make acrylic resins, and that is used in car industries as well as other purpose. On the other side, we have polyesters. Polyesters are used in is used in clothing, such that when we are going to have clothing, there are certain synthetic fibers which is basically the polyester. And on the other hand side, this is used in clothing purpose and for making washing tanks as well. Clothing and wash tankers. And there is another important thing which is about biopolymers. So this is something we have already covered in the organic in the biochemistry in 11th grade, so it's not much to worry about. This basically there are 16 bioelements, and out of those, six of them are the major ones, which includes the carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, and other 10 are basically also essential but not that abundant as the sixth ones. These are known as major elements, and alongside that we have some other elements which are present less in the body, which is known as trace elements. So all of these elements are necessary because they form the entire human body, and we're going to look into four major macromolecules which constitute the human body. The first one which we're going to look into is carbohydrate. These are hydrates of carbons, and they have the general formula CnH2n, CnH2o N, where the carbon to where hydrogen to oxygen ratio is 2:1, and that is basically also known as sugars or saccharides because they are sweet in taste. So these are known as sugars or saccharides. And an important fact about them is that important fact about carbohydrates is that they are found in both cyclic as well as open chain structure. And another important fact is that carbohydrates are divided into three major categories. Number one is the monosaccharides. Monosaccharides are having the general formula CnH2O, and that basically they are having carbon atom 3 to 7. They are having carbon atoms 3 to 7, and uh, they basically there are certain carbohydrates which do not have this ratio CnH2O. They do not have this ratio. Some carbohydrates do not have this ratio. For example, carbohydrates such as raminose. Raminose is a sugar, is a deoxyribose sugar. It's a deoxyhexose sugar. Raminose. Its formula is C6H12H12O5. This is raminose. And there is another sugar which we are most commonly known as deoxyribose sugar. So that is again having the formula C5H10O4. These are not following this rule. When you talk about the monosaccharides, we are going to have the general formula C6H12O6. But the structure would vary. Structure formula is complete is vary, but all of them are isomers. If you just look into first, we have glucose. Glucose is an it can exist. Now another important fact is the monosaccharides can exist in open chain as well as in cyclic chain. So if you just talk about their structure in human body, 99% of the monosaccharides, glucose and fructose exist in cyclic chain structure because cyclic chain structure is more stable than open chain structure as a result of less surface area available for attack, due to which they mostly exist in cyclic form while 1% of them exist in open chain form for the sake of let's say for the sake of for the sake of breaking down of purposes and respiration purposes. So anyways, this is an open chain structure, and on the other hand side this is the cyclic chain structure. Now on the basis of where the hydroxyl group is present, they also show optical isomerism, such that if they rotate they have optical isomerism such that if they rotate the if they rotate sugar, if they rotate the glucose, if they rotate plane polarized light towards the right side, they are known as dextrorotating glucose, D glucose, and if they rotate the plane polarized light to the left side, then it is known as the levo-rotating glucose or L glucose. So the artificial sweeteners are having L glucose in them, which are and because the enzymes in our body are D enzymes, and they cannot break down L glucose. due to which L-glucose goes inside the body, gives us a sweet taste, but does not digest in our body, does not absorb in our body, and is simply taken away. So, alpha D-glucose and beta D-glucose. This is another important concept. Whenever we are going to have OH group, and on the third carbon of the carbohydrates, on third carbon, if you can see, the third carbon is having OH group, which is present on the upper side. If the OH group is present on the upper side, then it means it is an alpha glucose. But if the OH group is present on the lower side, then it means then it is a beta glucose. So this is an important thing. Now let's just move on to the polysaccharides. Polysaccharides are again these are important parts. Polysaccharides, sorry, disaccharides, not polysaccharides. We first going to discuss oligosaccharides. Oligosaccharides basically are having when two or more than two monosaccharides combine together to form to form a dimer. It's going to form a disaccharide. The most common oligosaccharide is a disaccharide, and that is having general formula is C12H22O11. For trisaccharide, the general formula is C18H32 H32O16. Example of this is raffinose. An example of this is sucrose, which is also known as the common sugar or the table sugar. On the other side, we have a lactose, which is also known as milk sugar. On the other side, we have maltose, which is known as malt sugar. So these are some important sugars, and they must have uh, oligosaccharides need to have monosaccharides. Oligosaccharides need to have 2 to 10 monosaccharides in them. On hydrolysis, they yield 2 to 10 monosaccharides, but they cannot yield more than 2 to 10 monosaccharides. This is an important thing. And there's another important fact about oligosaccharides, that they can be hydrolyzed in the presence of a dilute mineral acid. So in that way, we can actually tell whether it is it is a disaccharide, a trisaccharide, or a polysaccharide. So in that way, we can actually obtain it. Now, when polysaccharides are those which are having more than 10 monosaccharide units, those are known as polysaccharides. Example of this is there are three main examples of these. Number one is starch. Starch is mostly found in the human body. It is found as a storage reservoir in plants. So it's pretty important, and that basically is not found in pure form. It is actually found in the form of a mixture of two different types of starch, which is number one amylose, unbranched starch, which all of these are polymers of alpha D glucose, right? And there is amylose, which is unbranched starch, that is around 10 to 20%, and there is amylopectin, which is around 80 to 90% of the starch. And amylose is unbranched starch, which is having alpha 1,4 glycosidic linkage, while there's another starch which we call as the amylopectin starch, which is a branching in it, and that branch is connected through alpha 1,6 glycosidic linkage. And this is present to 90%. Amylose is more soluble in water, amylopectin is less soluble in water, and amylose gives blue color in iodine test, and amylopectin gives red color in iodine test. So this is an important thing. Now, let's just move on to another type, which is animal starch. Animal starch, or we also call that thing as glycogen. Glycogen or animal starch, basically it is having a similar structure to amylopectin, but it is having more number of branching, and it gives red color only in the iodine test. So let's just move on to another one, which is cellulose. Cellulose basically is again, it's of all that we have studied, it is the most abundant carbohydrate. It is the most abundant polysaccharide on earth. Most abundant polysaccharide on earth is cellulose. And the pure form of cellulose is cotton, which is around 99% cellulose. And on the other side, more than 50% of the trees, woody trees, is cellulose. And this is another important thing, that cellulose has approximately 2500 units of the beta D glucose. Beta D glucose. And that beta D glucose is having the bond which we call as beta 1 for glycosidic linkage. And this is having a structure similar to amylose, except the fact that it is having beta D glucose instead of alpha D glucose. Alright, so this is all about carbohydrates. Let's just look into a quick overview of proteins as well. As how does protein design? Proteins basically are named as proteose because they are the first, which means the first priority in our body, because all of the structures, important structures in our body, are made up of protein. So if you just talk about a broad classification of proteins, they are divided into three major types. Number one type is the simple protein. Simple proteins are those which are only having amino acids in them. For example, let's say we are having legumin. Legumin is a protein, and there is collagen. These are the proteins which are most abundant in the animal kingdom. They form around 25 to 35% of the animal structures. These are the most abundant proteins in the animal kingdom, right? And on the other hand side, we have some other examples like keratin. We have examples like gelatin, keratin, casein. All of these are important proteins which are simply made up of amino acids. On the other side, there is a conjugated protein as well. Conjugated protein. Conjugated protein is the one. Conjugated or complex protein is the one which is having some other non-protein part with it, which is known as prosthetic group. For example, if you are having lipoproteins, lipoproteins are conjugated proteins. There is nucleoprotein. There is glycoprotein, mucoprotein, and in that way there are different types of proteins. On the other side, there is a derived protein as well. When we are going to derive a protein from these sources, then we are going to get a derived protein because they are derived from the simple and conjugated protein. So now let's just understand some types of protein based on the structure. So on the basis of structure, protein can be divided into four major types. Number one is the primary structure. Primary structure is the one which only involves the peptide linkage. There is no other linkage other than the peptide linkage, and that's a linear chain. This is observed when the protein is being made in the cell on the ribosome. And second type of protein is secondary structure, such that in secondary structure we have two major things, which is alpha helix structure and beta pleated structure. Alpha helix structure is in the form of uh, helical strand, and beta pleated sheet is like a folded sheet, a structure of the protein. These are formed due to hydrogen bonding. Hydrogen bonding, which is formed between the nitrogen and carbon of the amino acids, the carbonyl group and the amine group of the carboxyl of the amino acids. And then we have tertiary structure. Tertiary structure is very coiled structure. It's highly coiled because of the presence of disulfide bridges, because of the presence of ionic bridges, because there is ionic bonds, disulfide bonds, and there are a few other bonds as well. This is tertiary structure, and whenever this structure is also known as a globin. When we are going to have multiple globins combined together through hydrophobic interaction, then it is going to form a quaternary structure. It is going to form a quaternary structure. Example of quaternary protein is hemoglobin, which carries oxygen. An example of tertiary structure is myoglobin. So this is very important. Now let's just move on to some other structures, other important things, which is about lipids. Now, going through a quick overview of each and everything, the reason behind it is that we are not going. We have studied each and every of these, each and every concept from this topic in our previous classes. So if you feel overwhelmed by the things being taught in this lecture, you can surely look into the previous videos which can help you better understand the basics, and that's kind of a revision class. Alright, so lipids, we are not going to discuss the details, we are just going to look into some important reactions. Again, when you are going to perform hydrolysis of lipids, for hydrolysis of lipids we use enzyme which is known as lipase enzyme. This lipase enzyme, when it basically hydrolyzes lipids into glycerol and fatty acids. On the other hand side, there is an important concept which we call as an important concept which we call as saponification. Saponification is basically whenever we are going to take, let's say I take a lipid and I'm going to take a lipid and I'm going to treat it with sodium alkali or potassium alkali. Then it's going to produce uh, two things. Number one, it is going to produce glycerol, and number two, it is going to produce sodium salt of carboxylic acid, also known as soap. And that soap is used for making soap, right? And on the basis of this we can actually identify what we call as saponification number. What is a saponification number? Saponification number is simply the of number of milligrams of sodium hydroxide or potassium hydroxide which is required to saponify 1 mole of fat or oil. Then that milligram of sodium hydroxide or potassium hydroxide would be named as a saponification number. And that saponification number is different for different fatty acids. For example, if we are taking, let's say, I'm taking glycerol tripalmitate, right? I take glycerol tripalmitate. It is having, it requires 168,000 milligram of sodium hydroxide or potassium hydroxide to saponify 1 gram of glycerol tripermetate. This is the thing, and if you just, what I'm going to do is, in order to obtain a saponification number, I will divide it by its molecular weight. What is that molecular weight? It's something around 807. So when I divide it, I will get the answer which is near to 208. So 208 would be its saponification number. Some important significance of saponification number is that when there is an increased saponification number, there is uh, there are more number of short chain fatty acids. If there is a decreased saponification number, it means that there are long chain fatty acids. So this is used for telling the length of the fatty acids particularly. Now let's just move on to another important thing, which is thing which is catalytic reduction. We call it as catalytic reduction, hardening of oil, Sabatimer, Sendam reaction, catalytic hydrogenation, which we have studied a lot of times. We take nickel catalyst and then we perform hydrogenation, due to which we actually get a saturated fat. So this is an important thing. Now let's just understand a few other things, which is that whenever we are going to form, fatty acids, whenever you're going to react fatty acids with glycerol, it's actually going to produce triglycerides, which are the pure lipids. Again, like proteins, we are having three classes: simple lipids, pure lipids. There are simple lipids or pure lipids, complex or conjugated lipids, which are mixed with other types of lipids, and then we have derived lipids like sterols and cholesterol. So that's again something we have already learned. Now let's just understand a few more important things about few more important things, which number one includes iodine number. What is an iodine number? Iodine number is basically the number of milligrams of iodine which can be used to the number of milligrams of iodine which can be dissolved in 100 gram of fat or oil, that is known as iodine number. Iodine number basically tells us the unsaturation of a bond, such that when there is an increased iodine number, it means that there is a high number of unsaturation in the fatty acid. If a fat is having no double bond, then it means that it is having its iodine number as 0. Now, this is an important thing. Another important thing is about what we call as rancidity. What is rancidity? Rancidity basically is the foul odor or the characteristic smell that comes out whenever we are going to burn lipids. When you're going to burn lipids, we are actually when you're going to burn lipids or when the lipids decay, they actually give off a very foul smell, which is known as rancidity. Uh, and for example, if you just you might have noticed that whenever we are going to take a fish out of whenever a fish is being decayed or when a fish is being cut, there is a very foul smell coming out of the fish. The reason behind it is that the fish is having a lot, that marine animals have a lot of unsaturated lipids in them, and because of the unsaturated lipids present in the marine animals, when they are decayed, when they are taken out of water, they actually decay, and the unsaturated compounds when decayed, they give off a characteristic odor which is known as rancidity. And that's the reason that fishes have a lot of bad smell whenever we are going to buy them. We might see that the smell usually comes due to the decaying of the unsaturated bonds, and this is known as rancidity. And there's another important concept which is um, about the sterols. So sterols, basically, this is an again these are class of derived. These are derived lipids. Derived lipids. So we just look into the structure. The basic unit of sterol is a steroid nucleus. This is a steroid nucleus. This is the entire steroid nucleus. So it's the entire structure of cholesterol. Let me just draw a simplified structure. It's the structure of a steroid nucleus. This steroid nucleus is a 17 carbon ring, such that three rings are hexagonal, one ring is pentagonal, and there are total 17 carbon in it, except this R group. And basically, it is a fused ring, and that is having its scientific name is per hydrocyclo. This is a scientific name, IUPAC name, not the IUPAC name, but the common name, per hydrocyclopentano anthracene. This is the scientific name of this steroid nucleus, per hydrocyclopentano anthracene. So this is the entire thing. All right, now let's just look at some of there are three major steroid nucleus sterols present. Number one is the cholesterol. Cholesterol is present. There are 2 grams of cholesterol which is synthesized in our body, out of which 85% comes from the blood cholesterol and 15% from the dietary sources. And that cholesterol, I mean, can be harmful for our body in a way and can be beneficial for the body in a way as well, because lipids provide insulation. That cholesterol can help us resist cold temperatures, and on the other side, cholesterol can, on the other hand side, when it is in an overwhelming amount, it can lead to heart diseases as well. So this is an important thing about cholesterol. This is a basic structure of cholesterol, right? This is having a steroid nucleus, and this is the alkyl ring. Now, it is also used as a precursor for many different hormones. Like the steroid hormones are all having the precursor as cholesterol. And this is another important thing which we call as another important thing, and other very important thing, which is ergosterol. Ergosterol is found in fungi like ergot, uh, you know, uh, eh, you know, ergot fungi and yeast, and they basically, when it is treated with ultraviolet radiations, it is then converted into vitamin D2, also known as ergocalciferol. This ergosterol is found is released by ergot fungi, which ultimately causes the rye to become ergotted, and that ergot rye causes ergotism whenever it is eaten by someone, and that causes high amount of psychotic convulsions and dizziness and psychotic spasms, which is very dangerous and deadly. And now we have phospholipids. Phospholipids are an essential components of the human body. They are an essential component of the cell membrane. The cell membrane is made up of phospholipids, and if you just talk about the structure, it is like having a glycerol ring, a diglyceride, a diglyceride, which is having a attached phosphate group attached with a choline nitrogenous base, with a nitrogenous base choline. This is phospholipid. Now it's not necessary that every time we are going to have a choline. Instead of choline we can also have inositol, which is an amino alcohol. We can also have a serine, an amino acid. We can also have inositol, serine, choline. So this is about it. Now let's just move on to another important thing, which is enzymes. Enzymes basically is one that we have studied previously. I'm just going to mention few points about its action in order to just give you a revision of the things. So enzyme basically has got there are two major models on which the enzyme works. Basically, when enzyme acts on a substrate, it's going to first attach with the substrate to form an enzyme substrate complex, and then it forms performs catalysis, and then it forms an enzyme product complex. Then enzyme product complex releases the products, and then the products are released. This is how the mechanism works. There are two major models. Number one is lock and key model. Lock and key model. What does a lock and key model means? Lock and key model is one in which suggests that enzymes have a specific active site which is only specific for particular type of substrate, and it is due to this reason it specifies enzyme specificity. Second type of module is induced fit model. What does an induced fit model says? It says that the enzyme's active site is very flexible. It's flexible such that it modifies itself um, according to the according to the shape of the substrate, and this shows the this is a widely accepted model, and this shows the um, catalysis, the multiple functionality of the enzymes. So this is a model which is more accepted than the first one, but both models have significance. Both models are equally important. Nucleic acids, again, these are very important components. If we just talk about nucleic acids, we have DNA, we have RNA as the major nucleic acids. Nucleic acid, if you just talk about DNA, it was first discovered by Frederick Richard, and it was he basically discovered in those in the sperm of sperm cells of shark, and in the pus of white WBCs, and basically at that point he discovered DNA, Frederick Misher. So basically both of these components, this is known as deoxyribonucleic acid and this is ribonucleic acid. There are few differences between DNA and RNA. DNA and RNA has few differences. First thing is about basically the structure is like this. There is a ribose sugar. There is a ribose sugar, right? This is a ribose sugar. The first carbon is having a nitrogenous base attached to it. A nitrogenous base attached to it. It is for both DNA as well as RNA. Nitrogenous base attached on the first carbon, and on the fifth carbon there is attachment of the phosphate group. This is a structure of a, a very basic nucleic acid. So, if you just talk about DNA is having a nitrogenous base. DNA is having a nitrogenous base, which is purine, pyrimidine. Now, what are purine and pyrimidine? Let's just understand this. Purine is a nine-membrane ring in which four of the membranes are having nitrogen atom, and five membranes are having carbon atom. There are two rings in it. There is one six-membrane ring known as pyrimidine ring, and other five membrane ring known as imidazole ring joined together with each other, fused with each other. And this is purine ring. This is the entire structure. And if you just talk about purine, this includes example of adenine and guanine bases. And pyrimidine on the other hand side, it is a six membrane ring which only includes one ring known as pyrimidine ring. It does not have imidazole ring, and it is a six membrane structure having two nitrogen atom, four carbon atom. Now, if you just look at this structure, we have cytosine, thymine, and uracil. Now, DNA is having adenine, guanine, cytosine, and thymine. But in case of RNA, the thymine is replaced by uracil, such that instead of thymine, adenine adenine forms a hydrogen bond with uracil. This is an important thing. And second thing which I want to focus is on the number of oxygen atoms. Such that in the ribose sugar, in DNA, the ribose sugar is having one oxygen atom less than the original ribose sugar, such that one oxygen atom is less on the second carbon. On the second carbon, oxygen atom of OH group is converted into a hydrogen group, due to which it becomes a deoxyribose sugar. Becomes a deoxyribose sugar. Now let's just understand one more important thing, that this is having a ribose sugar and this is having deoxyribose sugar. Why does it happen so? Because the body requires D and A to be more stable, because DNA is essentially a carrier of genetic information from one generation to another. So it needs to be really stable, and in order to be stable, it needs to have it needs to give away it does not have any sort of oxygen on the second carbon, because the hydroxyl group present on the second carbon makes the DNA more reactive, and thus in order to decrease its reactivity, oxygen is not present on the second carbon, while ribose on the other side is not that ribose on the other side is not that stable. It is very reactive, such that after 30 to 45 minutes of the translation process, protein synthesis it does, it is decomposed really quickly. And then the other important thing is thymine is what? When I'm going to add a methyl group on the third carbon of uracil, I'll actually get a thymine. So basically, thymine is a more stable version of uracil. 3-methyl uracil is thymine. This is another important fact. And on the other hand side, we have what we call as phosphate group. Phosphate groups is the part which makes it pretty acidic, and that's basically what makes it acidic and what makes it actually a nucleic acid. So this is all about this video. Hopefully we'll meet again in another video with a new concept.